It's Dr. Von Mancha, and I'm with Ratio Christian, Mobile, Alabama, as well as Reasons to Believe, both apologetic organizations trying to concord science with the Bible. Today's talk is going to be on epigenetics, or does Darwin need a rethink? And on the middle portion of this slide, you can see the theory of evolution spelled out. The double squiggly line in red is a DNA molecule. And as you go below the DNA molecule, you'll see a gold star on the DNA molecule. This represents a mutation or a change in the DNA sequences. This is what the theory of evolution depends on: mutations, which cause a change to the body type. And if the body type is more fit, then this mutation is fixed by natural selection. Whereas epigenetics, which is shown on the far right, doesn't involve mutations. Instead, a tag, which is in purple, is placed on top of the DNA, and this tells the DNA whether to turn on or to turn off. In this fashion, the body types are also changed. Seven years ago, the Nature Journal, which is the most prestigious journal in biology and genetics and evolution, came out with this article called "Does Evolutionary Theory Need a Rethink?" When I saw this, I about fell out of my chair. I mean, at that time, evolutionary theory was supposed to be a fact like gravity and unquestionable. And these biologists basically were pointing out that this new field of epigenetics was changing everything about the way we thought. In the article, they made this mention because a lot of the older evolutionary theorists didn't like this. And so they mentioned that yet the mere mention of epigenetics often evokes an hostile or even emotional reaction amongst the evolutionary biologists. Too often, vital discussions descend into acrimony with accusations of muddled or misrepresentation. Perhaps haunted by the specter of intelligent design, the evolutionary biologists wish to show a united front to those hostile to science. In other words, their theory has been such a accepted fact for so many years that to come up with a completely different paradigm threatened them, and they attacked it. Instead of being welcoming of new information, they were trying to hold on to old, outdated information. Darwin wrote The Origin of Species back in 1859. He proposed that organisms naturally changed and that positive change made the organism more fit, survival of the fittest. And these traits were then passed on via natural selection. In his book, Origin of Species, which you can download for free, by the way, on Google Play Books, it says this under the chapter on natural selection, and he defines it in one paragraph. He said, if the conditions of life change and the form undergoes modification. Now, he didn't understand genetics back then, but he assumed that the species somehow undergoes modification, and that uniformity of character can be modified to the offspring solely by natural selection, preserving similar favorable changes. So this is a rather insightful statement, and later on, biologists incorporated this in with genetics and mutations. But during the same time, an Augustan monk by the name of Gregory Mendel did 2,500 actual experiments. Keep in mind, Darwin did no experiments. He was a naturalist. He did grow some plants and things and made observations, but he didn't do scientific method experiments. But Mendel proved that traits do not change like in Darwin's ideas, but that they're fixed. They don't morph together. In his case, he would mate green and yellow pea pods together, and he would get green or yellow. He didn't get a mixture of the two. Scientists rediscovered Mendel's work in the early 1900s. What happened was Mendel, being a friar, obviously didn't have the access to the better journals, and Darwin had a much better cheering section. So Mendel's work was basically forgotten about because of the journal that he published in. And then they rediscovered in the early 1900s and realized he was correct and Darwin was wrong. Fearing the demise of Darwin, they combined it with Darwinism to propose neo-Darwinism. Nice. So instead of giving it the credit to Mendel, which it really should be, which should be neo-Mendelism, they combined it with Darwin for philosophical reasons. Scientists philosophically preferred Darwin in a quasi-religious manner, as it proposed that we are simply the result of random nature. In short, we are just a meat sack with no free will. Mendel, on the other hand, showed that organisms are designed with discrete traits or factors. But Mendel was credited, at least, with being the father of genetics. In 1940, Julian Huxley, who happened to be a main leader of the euthanasia movement, proposed the modern synthesis, or what we call today the theory of evolution. The theory of evolution combined the concepts of mutation, explaining Darwin's natural variation, with population genetics and natural selection. So they became a field studying the different genes or alleles and their frequency depending upon the mutations that occurred. In the same year, Conrad Waddington, the father of epigenetics, said, "It is doubtful, however, whether even the most statistically minded geneticists are entirely satisfied that nothing more is involved than the sorting of random mutations by the natural selective filter." Conrad Waddington was warning the folks that were following the genetics and the mutations to tell them, said, mutations are not where it's at. There's many other things that cannot be explained by it, but they basically did not listen to him. In fact, there was quite a battle over this between the theory of evolution supported by genetics on the one hand and the naturalists who basically were studying epigenetics on the other. And he basically admitted in recent years it's gone strongly in favor of the former. This was a tremendous mistake and cost us many, many years of backwards research. Had we heeded Waddington's advice, we would have been 80 years ahead in the field of biology and, more importantly, medicine. That's how much evolution set us back. Not to mention the atheistic social Darwinist regime that murdered 100 million folks in the last century. It's the book on the left called Young Stalin, which I bought, and I'm going to show a quote out of it. But think about the guys up on the top right. All these people were strong Darwinists. They really felt like Darwin's theory fit with their political and philosophical views. In fact, Karl Marx, the creator of the communist movement, wanted to dedicate his book to the Origin of Species book. However, it was already out in print, so he wound up dedicating his book to Das Kapital, Darwin's book to Das Kapital. In the bottom right, we notice that all the 20th century atheist leaders were strong Darwinists. This is out of the Young Stalin book. When he was about 13, Leto took Stalin to a little bookstore in Gori, where he bought a copy of Darwin's Origin of the Species. Stalin read it all night, forgetting to sleep until his mom found him. "Time to go to bed," she exclaims. "Go to sleep. Dawn is breaking." Stalin said, "I love the book so much, Mommy. I couldn't stop reading it." As his reading intensified, his piety wavered. This book changed his life. One day, later in the next week, Sozo, which is Stalin, and some friends, including Grisha, were laying on the grass and talking about the injustice there is with rich and the poor. Then he amazed all of them by saying, "God is not unjust. He actually doesn't exist. We've all been deceived. If God existed, he would have made the world more just." "So, Krishna," yelled out, "how can you say such a thing?" And Stalin said, "I'll lend you a book and you'll see." He presented her with a copy of Darwin. So you can see at age 13, Stalin lost any sense of belief in a God of justice. And subsequently, the rest of his life was basically, he wasn't under any kind of judgment, didn't feel like he was under any kind of future judgment. So he could just basically do what he wanted because of his belief in Darwinism. Richard Dawkins, a famous atheist today and big time proponent of Darwin, says in a materialistic universe, there is at bottom no design, no purpose, no evil, no good, nothing but pointless indifference. And that's basically what these leaders in the last century had. It was all pointless indifference. They could murder millions and it really didn't matter. In 1953, Watson and Crick discovered the double helix of DNA. Combined with evolution and mutations, they felt it was an undeniable fact like gravity. In fact, that was coined from Richard Dawkins, the fact like gravity. For 50 years, evolutionists focused on mutations in the DNA alphabet of 4 letters, A, T, C, and G. A few scientists discovered there was a 5th letter, methylated C, but few listened until 20 years ago. This methylated C accounted for Waddington's epigenetics. Central to the theory of evolution was that mutations change the DNA letters. Example, A goes to T, which caused the resulting protein to be more fit. Natural selection would fix this change. In this little illustration, we can see the sort of stories that the evolutionists would have you believe. If you can see the jumping penguin, say for instance, this jumping penguin had a mutation which caused him to have stronger muscles in his fins so he could jump higher. Well, in that case, he would wind up finding the better looking penguins to mate with, and their kids would pass this trait on. Very simplified, but that's the way it's presented. In fact, Stephen Gould, the follower of punctuated equilibrium, who disagreed with neo-Darwinism and caused quite a stir, he said that all your stories are just those stories. And what he was referring to is Roger Kipling, the Jungle Book author, and he wrote a book called Just So Stories for Little Children. In other words, evolution in a way is for little children to understand. It is very popular because it is so easy to understand. But in fact, it's so simplified that it's in fact incredibly wrong. On your right, we see a little humor meme where the two camp guides are noticing how the bear is walking out with is walking out with a drill and a hammer and a crowbar. It said the bear-proof garbage containers have caused natural selection of a higher IQ. This is actually, even though this is funny, this is the sort of stretch that evolutionists would have us believe, because they can arm wave every story when they see a change in the DNA and say, well, in a billion years, this is going to be this. And it's kind of hard to argue with a billion year argument, if you will. In Francis Crick's model, he noted that DNA goes to RNA that goes to protein. Now, this is correct. However, this is only correct with 2% of the DNA. The rest of it does not follow this, and we'll discuss that later. In this case, on the top left, you can see a gold arrow called a DNA polymerase. These DNA polymerases, when they duplicate the DNA to another DNA, sometimes throw errors, and that does occur. But they had thought that these errors were just random for many, many years. In fact, really only in the last 5 years have we come to realize that epigenetics guides these random mutations to actually be biased in a critical fashion, not just random. So the DNA transcribes to the RNA, and then the RNA translates into proteins by tagging a bunch of different amino acids together. In your body, if you notice the middle pink portion, protein represents about 16% of the body. Now, it's actually spread out over the body. Uh, Water is the majority and fats are the rest, but it is the protein that makes that's made up of amino acids. And in the bottom right, you can see the different things that are made of protein: muscle, skin, organs, hormones, bones, nails, hair. So really the stuff that's making the body work is the proteins. Epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Now, this is different from neo-Darwinism in that the body is affected by the environment only, and if you had a different body, then it would survive in that environment differently. Epigenetics doesn't involve the change in the body. The different environment makes the body express things differently. It's a big difference. Changes that, and they change the way how your genes work. Unlike genetic changes, epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reads a DNA sequence. That's the difference. In evolution, there's a change to your DNA sequence. In epigenetics, there's not a change to the DNA sequence. There is a proviso, which I will discuss later. In the case of epigenetics, instead of the simple diagram here, where you just have DNA to RNA to protein, you have the chromosome, which tightly wraps up the DNA on the top left. This chromosome and the DNA has chromatin surrounding it, which is part of epigenetics. And then you have these basketball-looking balls called nucleosomes that tighten the DNA up. In the middle, you see a red and yellow tag. There are little tags that sit on top of these balls that control whether the balls open up or close, or how the DNA is expressed. Also, you can see with the unwound DNA, the red tags that are sitting on top of the DNA. These are epigenetic tags which turn the genes on and turn them off. Believe it or not, in 1925, the 5-methylcyclazine, which is that red tag, was first reported in bacteria. However, its biological importance was not intuitive for several decades, and that's because we got stuck in the theories that Francis Crick were proposing. We just didn't really think about it. After this initial lag, the ubiquitous presence of this methylated base emerged across all domains of life. In other words, it's not just in bacteria, but it's everywhere, and is revealed as a range of essential biological functions. This was discovered 25 years before the DNA molecule was even elucidated by Francis Crick. And we knew it was in bacteria, but why did they ignore it? I mean, this is 100 years before, 100 years ago. And these clues should have led us to study these things. In the case of epigenetics on the left, we can see that the environment shapes how the DNA is expressed. In this case, orange juice, grapes, kiwi, and different vitamins and nutrition modify epigenetically on top of the DNA to cause the genes to regulate, not change the gene sequence, but how it's played. And that then shapes the phenotype in the middle. The phenotype is the body, as you can see, or the outward expression of the cells. Whereas on the right-hand side, genetics and evolution involve DNA sequence variations, in other words, mutations. And this is proposed to cause gene regulation changes and change the phenotype. While this has been found on the micro level, this has not been found on the macro level. Discussing epigenetics and gene activation for improved health and longevity, you notice that there's things like on the top left, exercise, below that, nutritional factors, top right, environment, clean air, water, down below, emotional health. These all affect how your DNA is expressed epigenetically. We'll also discuss something later that's in the Bible. Per Darwin, for a mutation to be passed on, it has to be in the seed, the sperm and the eggs. Therefore, anything that happens during your life is not passed on because it's not in your sperm or eggs. Epigenetics, however, allows life's experiences to be passed on in what we call soft inheritance. And they typically transmit to the third and the fourth generation. This was also a Nature Journal article. Keep in mind, this is the top journal, and they came out with this article titled Epigenetics, The Sins of the Father. This basically blew up a lot of people's heads. I mean, they're basically realizing that the third and the fourth generation is exactly what was talked about in the Exodus, book of Exodus, chapter 20, and the Ten Commandments, and the sins of the father being the third and the fourth generation. Exodus 24 says, "Thou shalt not make for yourselves idol or any other likeness that is in heaven above or in the earth. You shall not worship them nor serve them. For I am the Lord your God, am a jealous God, inflicting the sins of the father on the children on the third and to the fourth generations of those who hate me." I used to always think this verse was just kind of God was unfair, that he was punishing the grandchildren, great grandchildren, for something the father did. It's not that at all. He's warning them not to do these things because it's going to affect, in essence, your genetics. And then he goes on to give a list of 10 commandments. And if you follow those 10 commandments, you're going to tend to avoid the stress and other things that can actually change your epigenetics. According to Darwin, the organism changes randomly, genetic mutations, and the resulting fitter organism passes its genes per natural selection. For epigenetics, the organism has the mechanisms to adapt, not evolve to the environment without changes to the DNA letters. Epigenetics, meaning above, directs tags placed on the DNA. Again, this was the diagram we saw earlier. In comparison, the left side, you see the theory of evolution. The DNA has a mutation represented by a gold star, and this changes the phenotype of the body. Now, this does happen, but keep in mind, we're discovering that epigenetics controls any mutations that occur to act differently. On the right-hand side, we see epigenetics, and a purple tag sits on top of the DNA. And this purple tag can be the tags on the unwound DNA or also on those nuclear balls called the nucleosomes. A little introduction to Epigenetics YouTube.
"78 organs and 206 bones, 600 muscles and 4,000 tendons, trillions and trillions of nerves. How do we..."
There are over 100,000 skin cells in 1 square centimeter of skin. Each of these cells contain close to 2 meters of DNA, which contain our genetic information. All the genetic material in our body is called a genome. Because our DNA is very long, cells package up their DNA together with proteins known as histones. The DNA and protein complex is known as chromatin. DNA is wrapped around histone proteins, forming repeated units of nucleosomes that look like beads on a string. Chromatin is condensed further to form a chromosome. We have 46 chromosomes in total, stored in the nucleus. The core of one nucleosome is composed of DNA wrapped around a histone octamer that consists of 2 copies of the major types of histones: H2A, H2B, H3, and H4. This organized DNA-protein complex allows the cells to regulate what genes are expressed and when. All the cells in our body contain the same DNA sequence. Skin cells, muscle cells, and liver cells contain the same DNA sequence. Yet all these cells have different structures and functions. The reason is that different cells only use or express certain genes. Within each cell, the DNA and also the histones can be tagged by tiny chemicals that modify gene expression. These chemical tags cause some genes to be turned on and some genes turned off. For example, muscle cells will have genes turned on that a muscle cell requires, and will turn off genes that a liver cell requires. Similarly, the liver cell will have genes for muscles turned off and genes that a liver requires turned on. These modifications are known as epigenetic modifications. Epi is Greek for above the genes, so modifications above the genes or on the genes. Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides: G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG island. And here the cytosine are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modifications. This is where acetyl or methyl groups are directly added onto histone tails, and this will modify gene expression. For example, histone 3K9 acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modifications, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
Okay, so you could see that was a good introduction basically to epigenetics with some more issues discussed. Our bodies have about 40 trillion cells in them, with each cell having about 6 feet of DNA, if you reckon.
Cole, he's my miracle child. So how does your miracle child get cancer? How does that happen?
Technical difficulties. Okay, where were we? Okay, so we have about 40 trillion cells with 6 feet of DNA in each. If you were to unwind that in space and place it end to end, it would actually go from the Earth to the sun and back 600 times. That's 93 million miles there, and then 93 million miles back. So almost a 200-mile round trip 600 times. If you took a spaceship going about 25,000 miles an hour, it would take it 900 years to travel it. Excuse me, 500 years to travel it. So you can see, because of this and the fact that epigenetics wraps up the DNA, that it is really totally in control. And this dwarfs the simplistic model of evolution that just assumes a DNA molecule out there. They presume the simple model of DNA to RNA to protein without really discussing the stuff that sits on top of it. This is the central dogma of Francis Crick, where epigenetics is far more complex and completely regulates the DNA to RNA to protein backbone. We are not just the sum of our DNA, rather the epigenetics plays the DNA to adapt, not to evolve. In this scenario, DNA is just an organ, whereas in the evolutionary model, DNA is everything. We dance to DNA. So what's an evolutionist to do? For 70 years, they've, quote, proved, unquote, DNA mutations accounted for natural selection. Epigenetics has no mutations. Faced with the fact that the mutational model is failing, they hope against hope to at least hold on to natural selection, since it's the buzzword that Darwin emphasized more than anything. Darwin didn't know about mutations. He just said things changed, and then natural selection fixed those changes. But it's not working because they cannot ignore 70 years of journal articles saying that mutations plus natural selection are working together. If you do a search on PubMed, which has millions of articles, including all the science journal articles going back for decades, and if you do the search of natural selection in parentheses, the result on the bottom left shows that there's 15,123 results. That means that there's that term natural selection is in over 15,000 articles, and many of these articles are proposing mutations and natural selection working together hand in hand because that was their mechanism. And unfortunately, it did work on a small scale, but not the kind of scale that they needed. However, if you look at natural selection epigenetics, there's only 300 results. A lot of these people are trying to say that epigenetics can work with natural selection, and that's still their buzzword, but it's just failing basically. Even though epigenetics works without mutations, there are mutations in some instances. However, epigenetics controls them as well. They are not random per evolution. One more time, Nature Journal, the most popular and prestigious journal of evolution today, came out with an article just last year, a year ago, saying why mutation is not as random as we once thought. I mean, this is also a nuclear bomb because mutations is what they base evolution on. In the article, they said random mutations are, and if you notice, this is bold, important part of the theory of evolution by natural selection. I mean, that's right. It's right there. That's it. Those two lines really says that this is the theory of evolution, mutations and natural selection, in which mutations give rise to adaptations that are passed on to offspring and alter their changes of survival. Then it says, since the first half of the 20th century, evolutionary theory has been dominated by the idea that mutations occur randomly with respect to their consequences. They're saying this has been going on for 100 years. We've been studying this way. This is the fact like gravity that's been shoved in our face for all these years. We conclude that epigenetic, and this is important, mutational bias, that's not random mutations, and we'll discuss it, what it is down here, challenges the prevailing paradigm of that mutation is a directionless force in evolution. Our discovery yields a new account, we're talking about the old is gone, this is the new account of the forces driving the patterns of natural variation, challenging a long-standing paradigm regarding the randomness of mutation. This basically says this does away with neo-Darwin evolution, and that we're no longer a directionless force as they philosophically loved 120 years ago. That's why they kept Darwin instead of Mendel, because Mendel didn't show directionless, he showed purpose. Darwin said, no, it's all directionless. We're just lucky. So epigenetics, mutational bias, which we'll discuss. In this diagram, again, of the epigenetic mechanism, you can see the top portion of the slide where the DNA is exposed, and on the bottom portion of the slide, when the epigenetic tags sit on top of things, the DNA is tightened up in the balls, the nucleosomes. By tightening up the DNA, first of all, you're able to get all that DNA in your 40 trillion cells that can stretch to the sun and back 600 times into the cells packed up tight. And guess what? They don't express themselves during these times when it's all tightly wound up. As well, they don't mutate, or when mutates do occur, they're reduced over 90% of the time. So 90% of mutations that occur are reduced as a result of this tightening up process of epigenetics. And more importantly, the mutations that do occur are guided by epigenetics so that it's not random. The exact letters that they want are picked. This is an example of the difference between epigenetics and mutations. In epigenetics, this is an article about the making of an ant cast. And this was published about a decade ago. Social insects represent a unique model of how the same genome or how the same stretch of DNA can give rise to entirely different phenotypes or body types. That includes soldiers of an ant, the common laborers, and the queen. New research on ants and honeybees also, because they're a social insect, points to DNA methylation, Lamarckian epigenetics. Lamarck is a gentleman that lived 50 years before Darwin, who described the process of the general processes which really equated with epigenetics. And this was 50 years before Darwin, and now they are starting to attribute to him epigenetics as well. And they say that these are the crucial factor for determining the caste of developing individuals. In this diagram, we can see that there's what's called a Waddington diagram, a Conrad-Waddington diagram. And this is how he proposed that epigenetics works. In the top middle, there's the omnipotent ball that rolls down, and depending on the different hills, it'll roll into different valleys, where it's unipotent or multipotent, and then finally the different body types. And on the bottom, you can see a minor ant left, in the middle a soldier, and a queen. They all look completely different. How can they come from the same stretch of DNA and look so different? If you put these guys in the ground for about a million years, you would think that their DNA is completely different. They're different species. Neo-Darwinism cannot explain this because mutations are so slow. You can't just mutate one group of the bodies, like the soldiers, from the queen. The same thing applies to Darwin's finches. If you recall, Darwin went on a ship called the Beagle, and he went to the Galapagos Islands, and there's a group of several islands in the Pacific, and he noticed that the beaks, the finches, had different beak sizes, and he reasoned that the different conditions on these different islands was such that these natural changes arose. He didn't know how, but once they did, they would pass them on through natural selection. And this is the icon of his theory, because this is the basic thing that gave him the idea of what he did in writing his book. However, when we studied the epigenetics, this has blown this completely out of the water. So evolution says mutations occurred and natural selection fixed those mutations for different forms of beaks. In other words, you should be able to find the mutations in the DNA. When they did the study on it, they compared the genetic changes that compared to the beak, that's related to the beaks, and then they compared it to the DNA mutations, and there were some mutations, right? But it was the epigenetic changes that made the change to the beak, not the mutations that made the change. In other words, there were very few mutations showing up in these beaks, whereas there was a lot of epigenetic activity going on. So, and this was done by Michael Skinner about 7 years ago. And I mean, it's like you've taken the heart of Darwin and stopped it basically because the finches was his big theory. Now we've shown that it's due to epigenetics. Well, you do recall about the discussion about overlooking epigenetics 80 years ago. Well, we also ignored 98% of the DNA as well because it didn't code for proteins directly. You know, you have to have the DNA goes to RNA, goes to protein, and only the protein changes the phenotype, which is under natural selection. If you don't have a change in the protein, it's non-Darwinian. And therefore, 98% of the body's DNA does not code for protein directly, and they called it junk DNA. So the Human Genome Project, which is led by Francis Collins, who is a Christian, he formed BioLogos, which is a Christian theistic evolution group to argue for evolution. Okay, so he applied evolutionary theory to his study in the Human Genome Project, and it took 11 years, $7 billion, a lot of resources, and the results came out at the millennium, and it was an utter failure because he only looked at the 2% of the DNA which they felt was under Darwin evolution. Oh, also, interestingly enough, we discovered the junk DNA 70 years ago. This is a lady who actually discovered it. Her name is Barbara McClintock. She was studying corn kernels. Have you ever noticed, you know, if you've been in a Cracker Barrel, they have a bin of corn, you know, the old-timey corn, and all the kernels have different colors to it. You know, you make it into a decoration. She observed, why is it that the kernels are not the same color? Because it supposedly has the same DNA. Isn't that a great question? It's right in front of you. she was the one that asked the question. So she started studying it and discovered that the DNA jumps around to different parts, that there's parts of the DNA that jumps, and she called them jumping genes, which were the equivalent of junk DNA, and that the way these genes, when they jumped around, changed the outlook of the kernels, and it could activate genes that are nearby to become active or inactive. Okay, she came out with this theory in her mid-30s, right? And everyone thought she was crazy. The Darwinists ignored her completely because this is not the way neo-Darwinism worked. They ignored her. They pretty much persecuted her because she left the field of research because she felt she was not getting anywhere. And then after the Human Genome Project failed, they said, hey, wait a minute, this lady, she already discovered this work 70 years ago. They said, well, we better do something about her. Well, they awarded her the Nobel Prize in Medicine as a result of it. And she's 83 years old there in that picture. We see a comic cartoon here. These are junk DNA in the garbage. He said, just because you don't understand us, you can't call us junk. Let's watch a real quick video on this.
In 2001, researchers of the Human Genome Project determined that only 2% of human DNA are genes, the sequences that directly code for the proteins our cells use to carry out their functions. The remaining 98% of the genome was concluded to be a biological wasteland. But it turns out that this supposed junk DNA is crucial for controlling gene activity, often acting as switches to turn genes on and off. These switches determine where and when a gene turns on. When a regulatory molecule binds with its corresponding switch on the DNA, it will cause the switch to flip and the gene to be activated. With the regulatory molecule bound to the switch, the gene will be expressed in greater amounts. When the regulatory molecule detaches from its switch, gene expression will stop. Typically, the gene will be surrounded by many switches that allow the gene to be turned on and off under the control of different regulatory molecules.
Okay. Okay, so again, what is the theory of evolution done for us? It's caused these guys to ignore 98% of the DNA. I mean, if you had any theory in science, say in physics or something else, and it told you to ignore 98% of the mathematical equations out there, would you accept that? Or would you question your own theory? These guys were so quasi-religious about their theory that they ignored 98% of the DNA. It's just unbelievable. And the head of the NIH, applied it in the same way. About within 10 years of the Human Genome Project, another group of folks called the ENCODE Project, and it consisted of 400 labs, and each of these labs had many people. So it was thousands of investigators looked at the 98%, and this article was published in Science. Science is not quite as prestigious as Nature, but it's a very big journal. And they say that this project, ENCODE project, writes the eulogy for junk DNA. Okay, junk DNA, the concept came up in 1970, and it took 40 years to get rid of that concept. In fact, junk DNA the idea was being spread around by Richard Dawkins when he would debate against creationists, and he would say junk DNA is the greatest argument there is against creationism because God created junk, basically. So this is, and now he's got his foot in his mouth. Here's Francis Collins on the left, head of NIH. He then, he was the head of the Genome Project, but he was also NIH head till about 6 months ago. He quit, and head of Fauci. I don't know if you heard, but there's some emails that have been circulating around where they knew that the DNA of the COVID virus came out of the lab, not out of the bat, and he realized it's time to get out of town. In any event, Collins was at a event in Chicago, and he threw in the towel about the junk DNA. He said, we don't use that term anymore. It's pretty much a case of hubris to imagine. No, he said it was hubris. It was hubris to imagine that we could dispense with any part of the genome. Why did he suddenly get this insight? Well, he got this insight after wasting 10 years and $7 billion. And I mean, 11 years. I mean, all the medical folks were waiting for these results. And it was based on his presupposition of evolution and evolution theory and this ridiculous concept that 98% of the DNA was useless. He just ignored it because of that. And now he's calling it hubris. Why didn't he realize it back then? Anyway, he says, as though we knew enough to say it wasn't functional. He's finally admitting it, at least. Most of the DNA we used to think was there just kind of spacer, turns out to be doing stuff. Well, this is after 10 years of sitting on top of a failed project that he finally comes out and admits this because of the ENCODE. They forced him to admit it, basically. What's fascinating is he wrote a book about DNA and God, where he was trying to promote the idea of God based on the fact that DNA existed, although not directly, because it was only promoting the fact that the Big Bang occurred and that caused the DNA. But in any event, he gave the same junk DNA argument as Dawkins did, because he didn't like creationists either. But he went in there and ripped that out of his book and republished it. I mean, that, You never do that. If you've made a mistake, you leave it out there. You make up for it in your next book and correct the mistake. But to pull it out and hide it is just, it's very bad. So medical researchers ignored the evolution of so-called junk DNA. Since then, researchers have been exploring the possibilities of the function of this non-coding junk DNA. They've discovered that they have some are producing small amounts of protein derived from this DNA, and they're starting to look into its roles. For example, junk DNA has been associated with heart disease and heart attacks. Heart attacks is the leading cause of death for men and women in most races and ethnic groups in the United States. One person dies every 34 seconds in the United States from cardiovascular disease. And yet it was junk DNA that probably holds the answer to this. And we have been ignoring it because of the evolutionary paradigm. In addition, cancer is the second leading cause of death after heart disease. The cure for cancer completely depends upon our understanding of epigenetics. You saw on the little YouTube that the gentleman said when the epigenetic tags are put on the wrong way, that can cause what's called epimutations, not the regular mutations in terms of letter changes, but the tags can get put in the wrong place, and that can cause cancer. And we know for a fact that this epigenetics guides the cancer causing situations. You remember the diagram I showed you? Let me see if we have that here. We have the diagram where the DNA is tightened up and then loosened because of the epigenetic tags. Well, what would happen, say, for instance, if you needed something to be expressed, but it accidentally gets turned off? Well, that's the sort of process that occurs with cancer. Genes that need to be on are turned off, and genes that are turned off get turned on by accident. And they're all controlled by epigenetics, and that's cancer. So that's another setback that evolutionary theory has given us. In this article in the Federalist, it discusses what's going on with epigenetics. It says that epigenetics is causing a major disruption in the field of evolution. As you can see from my talk, a recent news article says current estimates are that approximately 1/3 of professional academic biologists, and I would say it's above a half at this point, who do not believe in intelligent design and find Darwin's theories inadequate to describe all the complexity in biology. I mean, this is very straightforward. Everyone realizes this is stuff that we had no idea of. A new wave of scientists argue that mainstream evolutionary theory needs an urgent overhaul. Now, this is at a conference in London, and at this conference, people tried to boycott this conference based on the fact that it's going to be talking about evolution. Again, here are these scientists who don't want to discuss new facts, and they tried to boycott it. Still, a lot of people showed up, especially young people who were cutting bait on evolution. And Pigliucci was one of the leaders of the evolutionary thought, and he was doing a question and answer session in the conference. And one member commented that the disagreement between epigenetics proponents and the more conservative biologists, i.e. evolutionists, sometimes looks more like a culture war than a scientific disagreement. Pigliucci said, sure, it's a culture war, and we're going to win it. And half the room burst out cheering. I mean, this is unbelievable. To basically, okay, it would be like saying, okay, evolution is wrong, and half the auditorium burst out cheering. That didn't happen in the past. Those folks that are bursting out cheering are young people. They are cutting bait on Darwin. They're ready to go on to the new stuff because cancer research and health research is there. No, you can't find it in the other place. To tell you how bad it is, if you go back to PubMed and do a search on the theory of evolution, there's only 15 articles in the last 5 years on it. Whereas if you do a search on epigenetics, there's about 30,000 articles on it. It's just unbelievable, the difference. Peter Ward wrote a book on the left called Lamarck's Revenge, because he basically says epigenetics is changing our understanding of evolution in the past and present. He's a paleontologist and an evolutionist. He didn't necessarily want this to happen. And he makes this a remarkable quote. He said, but on occasion, even seemingly irrefutable scientific principles, that's evolution, and paradigms do falter because of new revolutionary discoveries that cannot be explained by the old theories, the old theory being evolution. And I mean, that says it there in one paragraph. We thought it was irrefutable, and now this new information's come out and it's blown it out of the water, basically. So evolution is like on the left, it's the walking dead, and everybody knows it, but nobody really wants to admit it. Join me at my Facebook group called Darwin versus Lamarck, and we will get to, as we cheer for the winning side and discuss these issues.